My name is Russell Foster. I'm Professor of Circadian Neuroscience at the University of Oxford, and I also chair the Nuffield Laboratory of Ophthalmology, and also I'm director of this new Sleep and Circadian Neuroscience Institute, and I'm here to give the public lecture. Well, circadian rhythms are essentially give us an internal representation of a day. What they're doing is fine-tuning physiology and behavior to the varying demands of the day-night cycle. So everything is being tweaked. So in anticipation of going to sleep at night, core body temperature will drop, metabolism will go down for the resting state. But in anticipation of activity in the morning, physiology is cranked up, blood pressure is cranked up, and we're then ready for the activities of the day. Yes, and in fact, I think it's enormously liberating and empowering because you can actually, my just as you go off to sleep, so my growth hormone will, now start to be peaking, and yes, I wonder what's going on with that morning rise in cortisol, that sort of thing. I, as an undergraduate, was fascinated with. I'll start again. As an undergraduate, I thought I'd become a marine biologist. I mean, I went to Bristol because I was a swimmer, and I thought I swim, therefore I've got to become a marine biologist. And then in my second year, I discovered physiology, and we were recording from the light-sensitive eye of a locust. And I thought the fact that you could record a change in electrical activity from this eye was just phenomenal. So there's that. And then also reading J.Z. Young's Life of the Vertebrates. Classic tome. I mean, just a brilliant book. And there was a section on lampreys and the fact that lampreys had this third eye, completely different from the lateral eyes. And I just thought this is so amazing and weird. And that really got me into how body clocks are being regulated by the light-dark cycle. And what's emerged is that there are broadly two groups of light sensing systems within the vertebrates. Those that build an image of our world, vision, of course that's what we're familiar with, but these other light sensors which build up an impression of the overall amount of light, irradiance, and they're using the changes in irradiance at dawn and dusk to set the body clock. And what's turned out to be really exciting for me is that these are different sorts of receptors using different sorts of light sensitive molecules, and I suppose that's really been what I've been doing for the past 20, 25 years. So the big changes, I think, in the past 20, 25 years has been the sort of the power of the approaches that you can apply to a research question. I mean, if we jump to the most recent paper that we've got now in press, we've been able to use RNAi techniques, RNA interference, to knock down the expression of specific genes and their protein products, and then look at the impact on the regulation of the clock. And in fact, a particular protein we've identified, when you knock it down, the effects of light on the clock are huge. So instead of taking 6 days to adapt to a new time zone, these mice were adapting within a day. So it's that power of manipulation of genes, gene expression, and then relating that back to the physiology, which I think is so exciting. And really, I suppose we are still, to some extent, technique limited, but boy, we can address so many more questions than we could when we were starting out, 20, 25 years ago. So why do I want to go on about 24-hour rhythms and these circadian rhythms and these body clocks? And I think it's because so much of the way we think about ourselves is of constancy, that we are the same creature throughout the 24-hour day, and we're not. We're fundamentally different. Our vulnerability to disease, our moods, our ability to process information are constantly changing. And beyond the really exciting biology underpinning that physiology are the real-world consequences. So, for example, we'll touch upon teenagers. Their body clock changes as we get older, from 10 to the age of 21. There is this genuine biological tendency to get up later and go to bed later. Now that has major consequences on the way we structure our educational day. And so I'd like to just flag up the fact that we are an extraordinarily dynamic creature, constantly changing, and that has real consequences in the way that we structure our society. Many thanks indeed, Louise. It's extremely kind of you. I think don't look too hard on YouTube, because there may be one example of me doing karaoke in Japan, which I hope never sees the light of day, but could. What I'd like to consider in the next sort of hour or so are the rhythms of life and what your body clock means to you, from eye disease to jet lag. In fact, I'm not going to have too much on jet lag, but hopefully there'll be time for discussion about jet lag right at the end. I've also been told to advance the thing. Let's just try that again. There we go. That a written version of this lecture, authored by myself and my good friend Leon Kreitzman, will be published in Experimental Physiology. Now, that's more of a reminder to me, I think, than to you guys, because I haven't even started the manuscript yet. So bear with me, Experimental Physiology. Right. Oh yes, British Neuroscience Association. Those of you who are watching online and general public in the audience, please be aware of the British Neuroscience Association. There's lots of resources on this website, not least links to brainfacts.org, which you may find interesting in getting further background information about the brain. Okay, let's kick off. What I thought would be fun to consider in the next sort of hour or so are the world of 24-hour rhythms. The fact that our physiology and behavior is constantly changing over the 24-hour day, and some of the consequences of that. And then talk about how those 24-hour rhythms are generated within the brain, and indeed touch on some of the biology of sleep and the importance of sleep. Then consider the role of the eye on locking internal time onto the external world, and the critical role that the eye is playing in this process, and the implications that information now has to clinical ophthalmology and eye disease in general. Then talk about some real-world examples, and we'll touch on shift work, but also want to talk about teenagers. Essentially, the theme of that bullet point is working against internal time. What are the consequences? And then finish with some, I think, really exciting new work in the area of mental illness and the relationship between sleep and circadian rhythm disruption and mental illness, and how that might arise, and some of the consequences of that overlap. Jolly good. So let's consider this world of 24-hour rhythms. And, of course, not to insult your intelligence, but we sit on a planet that is revolving once every 24 hours. And the consequences of that, of course, are huge changes in levels of light, temperature, availability of food, susceptibility to predators, you name it. And this complexity has driven an evolutionary response in almost all biology on the planet. We have adapted, we anticipate this predictable 24-hour oscillation. And as I was saying earlier with that terrifying clip, fine-tune physiology and behavior to the varying demands of activity and rest. And here we see some examples. Our mood, core body temperature, urine production, all of these components of our physiology are peaking at different times during the day, in the middle of the day versus the middle of the night. Now, what I want to stress is that if you or I went to a deep, dark cave, constant light, constant temperature, we'd still see changes over the 24-hour day in our biology. Our mood would go up and down, our blood pressure, all the rest of it. And the critical point is that these internal rhythms are locked onto the external day largely as a result of exposure to the light-dark cycle. And we'll talk about those mechanisms in a few moments. But let's now kick off and burrow in a little bit more depth into the biology of these changes. Here we see all of those parameters actually plotted. So we have a 24-hour time base here and a change in the parameter on this axis. And we don't have time to cover all of them, but I would like to consider just one or two. So core body temperature. Core body temperature shows about a degree variation over the 24-hour day. And in anticipation of sleep, core body temperature after around about 10 o'clock starts to drop, goes to its low point at around about 4 o'clock in the morning, and then in anticipation of getting up, core body temperature starts to rise along with metabolism. Now, that change in core body temperature is not only important for all our physiology, but has practical consequences if you're an athlete. I don't know, how many athletes in the audience? I can't see. Hands up. There's one. I'm afraid so you don't look very athletic, but I'll take your word for it. So what I wanted to emphasize is if you're a swimmer, this is some lovely data, and this looks at the early hours, one's ability to swim, one's speed, in the early training sessions at around about 6 o'clock in the morning versus the mid to late evening when the competitions take place. And what's extraordinary is that you can swim 100 meters, 2.7 seconds faster here than you can here. Now, that doesn't sound like a lot, but you'll know from the recent Olympics that 2.7 seconds is the difference between coming first and a really embarrassing toe curling last. And the point I want to make is that clearly, if you exercise, you're going to increase core body temperature, but it will be superimposed upon this endogenous, internally driven rhythm, this circadian rhythm in temperature changes. Let's look at blood pressure. Systolic blood pressure. We normally think of blood pressure, if you're lucky, as being, 120 over 70, 120 over 80. But actually, systolic pressure can drop almost half in the early hours of the morning, and again, in anticipation of activity, blood pressure will be cranked up for the demands of activity during the day. This change also has major implications upon health. So one's susceptibility to having a stroke is very much influenced by that internally driven rhythm. Of course, a stroke is a result of a buildup of a clot in the blood vessels going to the brain, and of course blood pressure is low, and in fact blood viscosity is higher during the night. So there's a greater tendency for these clots to build up during the night. They then become dislodged, of course, fly into the vasculature of the brain, cause a blockage, and parts of the brain, as a result of oxygen deprivation, will die. Now, this is some work by Peter Rothwell in Oxford, who's looked at time of day and frequency of having a stroke. And you'll see there's a marked change. In fact, there's a 49% greater chance of having a stroke between 6 a.m. and 12 noon than at any other time of the day. And of course, that is beginning to influence the way we think about when you should take anticoagulants. It would be smart to get those anticoagulants into the system here to dissolve the clots before this vulnerability zone kicks in. In fact, overall, there's a 29% greater chance of dying between 6 a.m. and 12 noon than any other time of the day. So congratulations, everybody. We've survived the most dangerous part of the day. And in fact, I'm now going to hopefully change your lives, because every time you look at your wristwatch and it's past 12 noon, so from now on you can think, phew, I've survived again the most dangerous part of the day. It's actually, when you get to my age, actually quite a relief. Let's now look at alertness. Alertness shows a really very marked change over the 24-hour day. And I think intuitively we'd all know how impaired we are in the early hours of the morning. But let's now look quantitatively at how bad we are at performing various tasks. This is a lovely study, and I just wanted to draw your attention to it. This is, on this axis here, a drop in brain function, if you like, cognitive performance. And in the first set of experiments, these guys compared the drop in performance, cognitive performance, against blood alcohol. So as you drink blood alcohol, there's a drop in cognitive performance. Now, I know that many of you won't know about that, particularly if you're a Brit in Ayia Napa. But there's, of course, a marked decrease in cognitive performance as you consume alcohol. At a blood alcohol level of 0.08 or so, you get a drop in performance of around about minus 15. And that's when you're legally drunk within the UK. Now, the same cognitive tasks were then explored over the 24-hour day. And so you see here that during the day, there's a bit of a dip in the middle of the afternoon. But after 10 o'clock, there's a massive drop in one's cognitive abilities. And what I think is so extraordinary is that one's ability to perform at, let's say, 6 o'clock in the morning is worse than if you had consumed sufficient alcohol to make you legally drunk. So if you take nothing from this lecture other than the fact that you're driving a car at this time of the morning, then you know your ability to drive that car is as bad as if you got into the car drunk. Now, in these particular experiments, the individuals were kept up throughout this whole period here. And so I can hear some people saying, what's nothing to do with the body clock? That just is fatigue. You're getting tireder and tireder. Not so. Because these individuals, of course, have been up 8 hours longer than these individuals, and yet their ability to perform tasks is increased. And it illustrates again this endogenous change in brain function over the 24-hour day. And just to show you the crash statistics, these are the crashes in the early hours of the morning, accounting for traffic density. So this really is, I think there's about a 5-fold greater chance of having an accident in the early hours of the morning than any other time of the day. Then I want to consider sleep. Now, sleep is perhaps the most obvious, the most marked of all of these 24-hour rhythms that we've been talking about. And yet, it's the one that we give so little thought to. It is remarkable how we disregard sleep. And so I'd like to place sleep into a slightly broader context in the next few minutes. First of all, I'd like to emphasize how much of our time is spent asleep. So this study from the US, the American Time Use Survey, showed that on average, 36% of our lives, our entire lives, are spent asleep. Which means that if you live to the age of 90, then 32 years will have been spent asleep. Now that clearly tells you something important, that this is an important bit of our biology. And you can compare the percentage of time spent asleep to other activities. So work-related, 16, watching TV, 11, housework, 8%. I have to say in my household, I don't think it's ever reached 8%. Socializing, I'm not sure about that one. And email, I would have thought that was closer to about 90% in my own experience. But what I wanted to emphasize here is the huge, prodigious amount of time that we spend asleep. And yet, we really don't think about sleep very seriously. And of course, our current attitudes towards sleep are in such contrast to the way we thought about sleep in the pre-industrial age. And I just thought I'd give you some quotes from Shakespeare. Absolutely beautiful quotes. Enjoy the honey-heavy dew of slumber, we saw in Julius Caesar. O sleep, O gentle sleep, nature's soft nurse, how have I frighted thee, from the Scottish play. And another Elizabethan. Oops, what's gone on there? I do not want that. Thomas Dekker. Sleep is the golden chain that ties health and our bodies together. Really prophetic. This is how people intuitively thought about sleep. They embraced sleep in the pre-industrial era. And yet in the 20th century, we really marginalized sleep. Here's the famous Thomas Edison holding one of his early light bulbs. And this is a real quote from Thomas Edison. Sleep is a criminal waste of time and a heritage from our cave days. Just amazing. And if we go further into the 20th century to the 1980s, that wonderful, gloriously awful, person, was it Gecko in Wall Street. And Wall Street was all about sort of, in fact, the strap line for Wall Street is money never sleeps. The implication being is if you sleep, you're resting on the job and you're not doing a sort of a decent day's work. And money never sleeps, along with, and I love this term, if you need a friend, get a dog. So we got to the low point in the 1980s, and I believe a famous politician once said, sleep is for wimps. I've actually tried to find a direct quote for that, but Margaret Thatcher was supposed to have said that. Okay, I'm pleased to report that we're beginning to move away from this really rather extreme position of sleep is for wimps and an indulgence and a luxury from the 80s to now. And we have to thank, neuroscience. Oh, bugger. Neuroscience. Yes, neuroscience research has begun to really give us some genuine understanding of the biology of sleep. So let's just pause and think about that for a moment. Why do we sleep? Now, I think it's very important to disentangle the evolutionary origins of sleep from why, as a species, we sleep now, and in fact, why different species do different things during sleep. I'm just really going to confine this argument to those mammals and perhaps birds with a very large brain. Okay, so why do we sleep? 3 ideas. The first is cellular restoration. Sleep is a process that allows the repair and replacement of key components of the body. And as an idea, it's been around since Aristotle. And it's gone in and out of favour. But it's vogue again because people have shown that a particular group of genes are only turned on during sleep, and those genes are associated with repair and replacement. So the idea that we're sleeping to promote cellular restoration is probably quite a good one in my view. Energy conservation. Sleep has essentially evolved to reduce energy demands. We sleep to save calories. I have to say I'm less convinced about this, because if you actually do the sums, if you compare the number of calories that you save during the sleeping state compared to sitting quietly and at rest, then on average you only save about 110 calories a night. Now 110 calories is the equivalent of a hot dog bun. And I would have thought that for such an extraordinary behaviour, 36% of our behaviour, it's got to be more than a hot dog bun. So I have to say I remain slightly sceptical about the energy conservation ideas. And the final idea is brain function. Sleep has evolved for brain performance, and particularly the laying down of memory, the learning tasks. And clearly, if you deprive individuals of sleep, their ability to lay down memories is smashed, really quite substantially. But there's more going on. It's not just laying down of memories and then recalling them. Our ability to process information, our ability to come up with innovative solutions to complex problems is enormously enhanced by a night of good sleep. And some beautiful data from Jan Bourne's lab has shown the importance of sleep in creativity and coming up with new solutions to complex problems. Okay, so what we've done in the past few minutes is consider this world of 24-hour rhythms and finished up with the most important of which, perhaps, or rather the most obvious of which is the sleep-wake cycle. And now I'd like to sort of burrow in and think about the mechanisms underlying these 24-hour oscillations. And of course, we go into the brain. Here we see a cartoon of the brain on its side. Here's the eye with the optic nerve going into the brain. And here we flip the brain on its back. Here's the spinal cord that will go down here, and the cerebellum, and the eyes going in from the optic nerve, and the optic nerve fuses, and this is the pituitary gland here, at this point. And that point is the optic chiasm. And if we were to take a slice through that level of the brain, we'd see two beautiful structures just resting where the optic nerves enter the brain. And these are called the suprachiasmatic nuclei, mercifully shortened to the SCN, suprachiasmatic nuclei. And in this genuine picture of an SCN, the cells have been labeled yellow, and the fibers from those, the processes from those cells have been labeled red. Now, we know, and I think this is an incredible observation made over the past 15 years, that individual SCN cells have the capacity to generate a 24-hour oscillation in electrical activity. So if we were to take an individual SCN cell out, put it in a dish, and record from it, you'd see 24-hour oscillations. And what that tells you, of course, is that the clock, fundamentally, is a molecular process. It's a subcellular process. There are, of course, about 50,000 individual clock cells in the SCN, and they work together to act as a master body clock, coordinating those 24-hour rhythms that we were just talking about. But I think what's so exciting is that the observation that a single cell can generate a 24-hour oscillation means there's a molecular clock. And what do we know about that molecular clock? And I would argue that perhaps one of the great triumphs in neuroscience over the past 20 years has been an understanding, a fundamental understanding of what makes this clock tick. It arose from studies that started in the fruit fly, Drosophila. Genes were identified in the fruit fly that were linked to the 24-hour rhythms of these flies. 1970s. Nothing much happened until the sequencing of the genomes, human genome and the mouse genome. And very similar genes that had been identified in the fly that regulated the body clock or generated the body clock were found in humans. And when those genes were mutated, and looked at in a mouse, you saw very abnormal 24-hour rhythms. And so what became clear is that the molecular clock in us and in mice, and indeed in all of the animal world, is broadly conserved. And that also, I think, is remarkable. Species separated by perhaps 600 million years of evolutionary divergence are still building their body clocks in largely the same way. And so what does that clock actually consist of? Well, what you've got is a molecular feedback loop, and I'll just sort of illustrate what I mean in this cartoon. You've got a bunch of clock genes, and there are around about 12 to 14 of these genes that have now been identified as playing a key role in the generation of these 24-hour oscillations. The clock genes are activated, and so they produce a message, which is then translated into a protein. These clock proteins then interact, but then enter the nucleus and turn off their own genes. You turn off the genes, no more message. The proteins in the cytoplasm are degraded. The proteins in the nucleus are degraded. And the clock genes are then liberated and can produce more message, more protein, more inhibition. And what that does, that molecular feedback loop, is essentially produce a beautiful rhythm of protein generation and degradation, and it's that oscillation that essentially drives, coordinates rhythmic biology in the rest of the organism. Now, changes in these clock genes are being linked to particular morning and evening types. So here's a cartoon of a time base here, it's about a day and a half. And you see this is the sleep episode here. People are going to bed at around about 11 o'clock and they're getting up at around about 7 o'clock. This is an idealized. Very few people have such good normal sleep. And you'll see here that at the weekends there's a tendency to go to bed a little bit later. You know, this is a social animal, of course, having a bit of fun at the weekends. What you also find, though, are individuals who are massively delayed. So they're getting up very, very late and they're going to bed very late. And that's even further exaggerated over the weekends. And there are individuals who are going to bed at around about 7:30 in the evening and getting up at 4, 5 o'clock in the morning. In fact, there's a very, very nice study with a condition called familial advanced sleep phase syndrome. These individuals are going to bed at around about 7:30 in the evening and getting up at 4 o'clock in the morning. And what's turned out to be, I think, truly remarkable is that there's a single change in one of the clock proteins which affects its stability. In fact, what it does is make the clock speed up. And so these individuals, their whole clocks have been sped up in time. But the point I'm trying to make is that morning and evening types are not simply something that we adapt to, is that we are to some extent driven by our genes. And another myth I'd like to dispel is this one, Benjamin Franklin, who said, he didn't say that, and I hope that'll be taken out of the thing. There we go. Oh. How can I turn these things on without? No. Help me, somebody. I need to advance this without trigger. Thank you. Early to bed, early to rise makes a man healthy, wealthy, and wise. This statement is wrong at so many different levels, of course. And I would like to emphasize that there is no evidence that those morning people who get up early and go to bed early are richer, smarter, have bigger cars, bigger houses, are more pleasant. There is no evidence that morning people are nicer than evening people in any way, shape, or form. And in fact, in my experience, the only difference between morning people and evening people is that morning people are just horribly smug. Okay. So, what I've sort of said so far is that we've got this wonderful master clock, the SCN, ticking away with all these individual cellular oscillators. And I haven't emphasized it, but I've sort of implied that the light-dark cycle sets all of these individual oscillators to the real world, to the external world. And then, again, what I've sort of implied is that the master clock then forces 24-hour rhythms on the rest of the body. But what's turned out to be, again, a remarkable discovery is that that is not correct. The master clock does not force 24-hour oscillations on the gut, on the brain, on the liver. But each cell in the body has the capacity to generate a 24-hour oscillation. Real big change in the way we thought about the organization of the body clock system. What we thought is that there was a signal here that just imposed rhythmicity. Now we understand there's a signal here from the SCN which is coordinating billions and billions of individual cellular oscillators so that internal time is appropriately coordinated with the external world. So you can think of this structure here as rather like the conductor of an orchestra, producing a regular temporal beat from which the component parts of the orchestra, the cellular oscillators of the body, take a reference cue and align their rhythms accordingly. Now, if you shoot the conductor, then that organizing sort of signal is gone and the various oscillators, the various parts of the orchestra drift apart. And instead of a glorious symphony, you then have this cacophony of noise. So you have this incredible hierarchy of organization. But the fact is that every cell in the body probably has its own molecular clock, as far as we can tell. Now, there's a lot more, though, to sleep than just this body clock. And I wanted to emphasize the complexity of sleep, because it's something we'll come back to right at the end of the talk when we talk about mental illness. Here we have the rest-activity cycle, and it's absolutely true that the body clock is critical in adjusting all the components of the sleep-wake cycle to the light-dark cycle in the external world, to the day. And what's happening is that the body clock is saying now is an appropriate time to be awake, and now is an appropriate time to be asleep. But it's not the only driver. There's another very important component that has been called the homeostat. And in fact, it's the intuitive part about sleep. The longer you've been awake, the greater the sleep pressure, the greater the need to sleep. And the reason you don't keep on falling asleep during the day as the sleep pressure builds up and up and up and up is because it's counteracted by the clock saying, stay awake, stay awake, stay awake. It's only at around about 11 o'clock at night when the sleep pressure is really high and the clock says, yep, now is a good time to go to sleep, you go into consolidated sleep. What is this homeostat? It's rather complicated. We know that a substance called adenosine seems to be really important, and a buildup of adenosine is part of the buildup of that sleep pressure and the need for sleep, which explains, of course, why caffeinated drinks keep us awake, because they block those adenosine receptors in the brain, and they override, to some extent, the need for sleep by the buildup of adenosine and the sleep pressure. Of course, as we sleep, then the sleep pressure winds down. And so there's a very important sort of loop here relating to the sleep-wake cycle. In us, of course, social timing, the alarm clock, is often forcing a rest-activity cycle upon our biology. And we'll talk about that within the context of shift workers later on. But of course, the sleep-wake cycle will in turn feed back upon the social timing. The sleep-wake cycle itself has a big effect upon our mood, our cognitive abilities. We've already touched on that. If you don't get sleep, your ability to process information is impaired, and indeed our mental health. And of course, these factors feed back upon the sleep-wake cycle. So we see a high order of complexity, which is again an issue that I want to come back to towards the end of the talk. The other important thing to absolutely emphasize is that unlike the body clock, which is this sort of single structure within the brain, which is so critically important, sleep doesn't arise from a single structure, but is a network property of the brain. And let's just look at that for a moment. Here's a section through the brain. Here you see the spinal cord going up into the basal brain. And we know that structures in the basal forebrain are really important for sleep. There are at least 4 key structures within the hypothalamus: the lateral hypothalamus, the suprachiasmatic nuclei, the tuberomammillary nuclei, and what's the other one? Oh, anyway, there's 4. And all of these are interacting to regulate a whole raft of structures in the mid and the hindbrain. So we've got big chunks of the ancient brain very much involved in the development and the regulation of sleep. These all interact, feed forward to the cortex, and essentially keep us awake or put us into sleep. And the other really important thing to emphasize is that it's not just one brain neurotransmitter involved in sleep, but everyone you can possibly think of. So you've got glutamate, GABA, ghrelin, orexin, also called hypocretin. It's this defects in this pathway that have been associated with narcolepsy. If you don't produce enough orexin, then there's this tendency to fall asleep during the day. We've got noradrenaline, histamine, antihistamines. If you get the drowsy antihistamines from certain chemists, then what they're doing is inhibiting the ability of histamine to keep you awake. You've also got dopamine, and you've got serotonin, acetylcholine. The point I want to make, and please remember this image because we'll come back to it later, is that this complexity makes the sleep-wake behavior very vulnerable to disruption. Okay, so let's now move to our third topic, which is the eye, light and time. This is a sort of cartoon I've shown you before. The master clock, eye, sets the clock to the external light-dark cycle, and here we see a rest activity profile. Each line represents a day. In this individual, they're awake, and this is when they were asleep, and it's beautifully aligned. Now, I just want to emphasize that if you destroy the SCN, if you have, for example, a tumor in this area of the brain, and we've actually studied this in certain patients, then as the tumor progresses through the anterior hypothalamus and destroys this critical part of the brain, then you lose these beautiful 24-hour rhythms and you have a completely non-24-hour pattern. All of those rhythms we were talking about earlier are smashed, they're lost. But I'm going to be focusing in upon the eye. What happens if by some ghastly accident you've lost your eyes, you have no eyes? Then you lose your ability to align the clock to the external world. The clock, of course, keeps on ticking, but it's no longer exactly 24 hours. The body clock of humans is slightly longer than 24 hours. And so if we were to go into complete darkness, or if we'd lost our eyes, then we'd get up later and later and later and later each day. Now, the thing that is, I think, so fascinating is that we're expecting this wonderful, beautiful structure to perform two very different light-detecting tasks. Now, the familiar function of the eye, of course, is image detection. It provides our sense of pattern, it provides our sense of color, it's the way we sense much of our world. But the detection of images is a fundamentally different sort of light-detecting task than is the sort of thing required to regulate the clock. What the clock needs is an overall impression of the amount of light at dawn and dusk. It needs a measure of environmental brightness. And the big problem I had when I started getting into this was, how can the eye, which is an exquisite image detector, also detect the overall amount of light in the environment? If you think about image detection, for it to work, once you've grabbed light, you've got to forget you've seen it within a fraction of a second. Otherwise, of course, our world wouldn't be a series of crisp images. It would be a complete blur. And so the problem was, how can an image detecting system also reliably extract brightness and hence time of day from the external world? If we look at the eye, and this is the sort of a textbook image that many of you will be familiar with, here we have the eye, and we're taking a little section through here. Here's the classical photoreceptors, the rods and the cones, the rods providing us with our dim light vision, and the cones working together to provide us with our sense of color. And the first stages of image detection is processing of this information from these rods and cones in the inner part of the retina. Horizontal cells, amacrine cells, bipolar cells. And then collectively, these have made some sort of decision, so that by the time we get to the ganglion cells, we get a spike, an electrical change, which is then sent along these axons of the ganglion cells which form the optic nerve, and they project to the visual centers of the brain. Okay, so the big question is, how can those cells extract brightness information from the environment? And what we did was to take actually different mutant mice, mice with hereditary retinal disorders, mice in which the visual cells, the rods and cones, had largely but not completely degenerated, leaving rudimentary, rather poorly preserved, in this case we see a poor cone photoreceptor. The point being, though, that with this huge insult to the retina, they had no image detection that we could assess in any way. They were visually blind. We then decided to take these 3 blind mice, and see how they run, and put them into a running wheel. And the assay was very simple. If they could see the light-dark cycle, despite this massive visual loss, then they would align their activity to the light-dark cycle. Remember, they're nocturnal, so they'll be active during the dark, and they'll be asleep during the light phase. If, however, that loss of rods and cones had smashed the ability to regulate the clock by light, then we would see this sort of pattern where the mice would sort of show a different activity onset day after day after day. So what happened with those blind mice? Well, they showed no impaired capacity to align their rest activity cycle to the light-dark cycle. And what was really exciting was that these mice were visually blind, but not clock blind. And we knew it was the eye, because if you use sort of opaque contact lenses and you cover the eye with an opaque contact lens, the ability to regulate the rest activity cycle is gone. They would drift. So it's definitely something within the eye. These are early experiments in the 90s, and on the basis of these, we sort of said, well, hang on, maybe there's something else in the eye. Maybe there's another light sensor, a novel, unrecognized photoreceptor system within the eye. Now, that was 20 years ago when I was considerably younger, and the response from the vision community at that time is summarized in the following slide. It was ferocious. I remember giving a talk in the States and presenting the early data, and somebody at the back stood up, said, "Bullshit," and just walked out. The arguments why this was such a completely stupid idea are sort of summarized in the next few slides. The first, which is not an argument at all as far as I was concerned, is that we've been studying the eye for 150 years. Are you seriously telling us that we've missed an entire class of photoreceptor within the eye? Well, that wasn't a sensible argument. Yes, it's perfectly obvious that we've missed another receptor as far as I was concerned. A more constructive, well, actually, it was I suppose, a semi-reasonable criticism, was that the circadian system, these 24-hour body clocks, can be regulated normally with just a few rods and cones. It's a really simple task, so you only need a few degenerate rods and cones left for it to work perfectly normally. Good reasons why that wasn't the case. However, what we had to do, of course, was better experiments. It's true, we didn't have the perfect model. We hadn't got the perfect result. So what we did was then generate a mouse which was carrying a naturally occurring retinal degeneration coupled with a transgenic animal which had lost all of its cones, and this had lost all of its rods. So we absolutely had no rods and cones in this eye. And to our intense pleasure, these mice were able to regulate their rest activity cycle absolutely normally. There had to be another receptor system within the eye. And let me stress, if you prevented light from getting to these eyes, then this wouldn't happen, the rhythm would drift. That's mice. What about humans? Well, we were extremely lucky to work with some ophthalmologists, and they identified a particular lady, in fact, who had no rods and cones, and she had no light detection for 50 years, complete loss of all of these rods and cones in her eye, and she also showed normal 24-hour rhythms in behavior, beautifully aligned to the light-dark cycle. So where we had got to was, from both humans and mice, is that in the absence of the rods and cones, you could regulate the clock perfectly normally. There's got to be something else within the eye. Was it horizontal cells, bipolar cells, amacrine cells, or these ganglion cells? And we now know that about 1 in 100 of the ganglion cells are directly sensitive to light using a novel light-sensitive molecule and a new photopigment. These are called photosensitive retinal ganglion cells. Okay, the point is, and this is what they look like. They are just beautiful cells. Here's the cell body, and these are the processes, and we know that these processes are directly light-sensitive. The light-sensitive pigment is embedded within the membranes of these processes here. And in fact, if you look at the broad arc of the eye, this is just a segment of the eye, but you see that there are loads of these cells, about 1 in 100 of the ganglion cells are light sensitive, and they sort of form a photosensitive net that encompasses the arc of the eye. So as light goes through, it's detected by these cells, and they grab this light and then fire that information in to the SCN, the master clock. Now, what these data meant, of course, is that visual blindness need not result in the loss of all light detection by the eye. And why that's important is because sleep and 24-hour rhythms abnormalities are almost ignored entirely in clinical ophthalmology. And I want to give you some sense of where we're going with this information. And I'm very lucky to work with clinical colleagues in the Oxford Eye Hospital, notably Susie Downs. And we've been asking the question, what's the impact of ocular disease on human sleep-wake biology? And I'm just going to summarize some of the key points here. Well, the first point is that there are many diseases in humans which result in the loss of these rods and cones. So, for example, diabetic retinopathy, Leber's congenital amaurosis, and choroideremia, and there are many, many more. The point is, of course, is the rods and cones are largely lost, and these individuals may be visually blind, but those photosensitive ganglion cells are still there, and they're still functioning. And such individuals, therefore, need to be encouraged to expose their eyes to sufficient daytime light so that their body clock will align to the light-dark cycle. The advice by many ophthalmologists is that, well, you should wear dark glasses, and there's no often good clinical reason for that. It's just that the eyes may look rather nasty, and so they're not very appealing for the rest of us to look at these individuals. So dark glasses, of course, would block out that light for the regulation of the clock. Even more terrifying, and it has happened, but it's less so these days, is that the argument would be, well, you're blind. You can't see. You can't see to look after your eyes. Your eyes are a source of infection. So I tell you what, let's make life so much simpler. We'll take those dead eyes out and pop in one of these. And of course, the implication of doing that, completely unwittingly, is that you may then be removing the eye with photosensitive ganglion cells and therefore plunging individuals who are already blind and have all the appalling consequences of loss of vision, but then applying, sort of imposing a world of timelessness on these individuals, essentially giving them unremitting jet lag for the rest of their lives. The other situation, of course, is that there are diseases of the eye which destroy the inner retina, including those ganglion cells, such as advanced glaucoma, where even though the rods and cones may well be intact, there's no way in which the rods and cones can talk to the brain and give you vision because the optic nerve has been destroyed. And of course, in the process of destroying the optic nerve, you'll destroy those photosensitive ganglion cells. But again, all is not lost. These individuals need to be advised about taking certain treatments and agents that will consolidate the sleep-wake cycle. So what I'm trying to say here is that the clinical diagnosis of complete blindness must assess the state of both the visual system and these photosensitive ganglion cells. Eye loss, of course, plunges individuals into a world that lacks both vision but also any sense of time. Clinical advice in ophthalmology must incorporate the impact of eye diseases on sleep and circadian timing. The world of the ophthalmologist has become doubly complicated. They've had to deal with vision, of course. Now they have to deal with sleep and internal time. And the numbers that could be affected by all of this are truly huge. 39 million people worldwide are blind, completely blind. 285 million show severe visual impairment, and 246 million show a limited visual capacity. And the extent to which a knowledge of these cells could improve the quality of life of almost 500 million people worldwide, I think is really important and really exciting and must now be taken into account. So what we've seen, I think, with this topic is a naive set of questions. How is the body clock regulated by the eye? Has now led to the discovery of a new receptor system within the eye with major clinical and applied consequences. Let's now look to other applied consequences of knowledge about the clock. And this is sort of a shorthand for the impact of a mismatch between internal and external time. This is a cartoon again of days, and this is the ideal good sleeper. They're going to bed a bit later at the weekend, but broadly speaking, they're going to bed at around about 11 o'clock in the evening and getting up at around about 7 a.m. It's quite rare, I have to say. The elderly. So what you see in the elderly is a tendency of very reduced sleep and fragmented sleep. They're going to sleep a little bit earlier, but they're waking up much earlier, with several episodes of waking up during the night and also periods of sleep during the day. One of the main problems, and one of the things you hear so often, is that I wish my sleep were better. And we have almost no idea at all why sleep in the elderly is so disrupted. It's an incredibly important area of future investigation. I flag that up for your attention. But I want to move to teenagers. So what teenagers are showing is a greatly delayed and reduced sleep. So they're going to bed very, very late. Weekends, it's even worse. And then, of course, at the weekends, they're sleeping in until midday. Many are having a nap in the middle of the afternoon or late in the afternoon when they've got home from school. Now, what's going on in these teenagers? There is genuinely a delayed body clock from about the age of 10 to 21 and a half in males, 19 and a half in females, a tendency to go to bed later and later and later. And that turns as you get from your early 20s back down to your 50s. And we don't understand why, but it's probably due to the changing steroid hormones, the sex steroids like estrogen and testosterone, which are changing very dramatically as we age. So part of this tendency to go to bed late in teenagers is genuinely due to biological phenomena. And so all those rhythms that we talked about earlier, which I sort of was referring to as sort of the average adult, are delayed by at least 2 hours on average in most teenagers. And let's look at some of the consequences of that. So here's the 24-hour alertness pattern that we looked at, and here's our healthy adult. And remember, alertness begins to rise in anticipation of activity, and we are at our most alert at around about 10 o'clock in the morning as adults. Teenagers, on average, are 2 hours delayed, so they're getting fully alert by 12 noon. And in around about 10 to 20% of teenagers, they are delayed not by 2 hours, but by 4 hours. So they're not showing their peak levels of alertness until 2 o'clock in the afternoon. Now, that means we've also got reduced sleep as well. And so what's happening is they're going to bed late, but the alarm clock is getting them up in the morning, and so sleep duration is horribly affected. It's further compounded by this tendency to play games in the bed. Some very dodgy practices going on in teenage bedrooms. I mean, you've got all this gaming going on. Oops, nope, I don't want that. You've got texting until the early hours. You've got watching television until the early hours. And so this biological predisposition is enhanced and exacerbated by all the ways in which now teenagers behave with all of this technology in the bedrooms. The bedroom is not a place of sleep, but it's a basis for entertainment in many cases. Mary Caskoden, who's done some amazing work in this area at Brown, suggests that on average US teenagers are getting about 7.5 hours a night of sleep, but as many are getting 6.5 hours. What she says is that to be optimally alert for full brain performance, teenagers need 9 hours of sleep every day. And many, many don't get anywhere near that. Does it matter? Well, okay. This is some studies by David Goldstein and Lynn Hasher. And what they did was look at the correct answers for various cognitive tests, either mid-morning or mid-afternoon. And perhaps no great surprise is that the teenagers showed a 10% improvement from mid-morning to mid-afternoon. That fits with the biology we just talked about. What's very interesting, of course, is that the adults, their teachers, went in the opposite direction. And this is the problem. I know when I talked about this a few years ago, many teachers contacted me and said, God, you know, living in your ivory towers in Oxford, you don't have the first bloody idea about what it's like. I know when I can teach the most demanding classes to my students. And it's the morning. And of course, what's happening is the teachers are coming in, they're feeling bright, you know, optimal alertness by 10 o'clock in the morning. The kids are half asleep, so they're easy to control. By mid-afternoon, the kids are bouncing off the walls because they've finally woken up. And of course, the teachers are feeling particularly jaded. So we have a real dilemma here, that the biological rhythmicity of our teachers and our teenagers are not ideally matched in the educational sector. But what's the impact of a later start day for school times? And of course, what we did was have a later start time coupled with what we've called sleep hygiene, and I'll explain what I mean by that. So these are from Monk's Eton School, and a terrific headmaster, Paul Kelly, who really implemented all of this work and is responsible for these data. And what he did was have an 8:50 start in 2010, and then shift that to 10 o'clock in the morning in 2011 and 2012. And then he looked at the impact on students reaching the minimum government standards in terms of GCSE passes. 35% of the kids were getting the minimum government standards with an 8:50 start, but with a 10 o'clock start, that jumped to 53 and 52%. This was a school that still, unfortunately, fell below the national average. However, the biggest jump seemed to be in those kids who were socially disadvantaged. 12% were getting the minimum government requirements, but with a later start, 42 to 42%. And they actually then were above the national average. Now, these are relatively small numbers, and I wouldn't want to push this too heavily, except for the fact that these data are entirely consistent with the sort of thing that's been found by Mary Caskoden in the United States, and indeed with researchers in Germany. So there is a real impact, I think, of starting the school somewhat later in the day. But it's got to be more than just a later start to the school. Otherwise, the kids will just go to bed later and later. They must also need to know how important their sleep is to them. And this is a piece that we wrote fairly recently, and I draw your attention to it, 22nd of April this year. And what we try to emphasize here is, okay, well, how much sleep do you need? Well, you know how much sleep you need if you're dependent upon an alarm clock or parent to get you out of bed. You're not getting enough sleep. If you take a long time to wake up, if there's lots of sleep inertia, you can't get out of bed in the morning, you're not getting enough sleep. If you feel sleep irritable during the day, and if your behavior is overly impulsive and you're doing stupid things, you're not getting enough sleep. What do? Well, you need to take certain levels of control. The bedroom must be dark and not too warm. It must be a place that encourages sleep. Don't text, watch TV, use computers for at least half an hour before you go to bed. Start to wind down. Avoid bright lights at least 30 minutes before you go to bed. Now, this is extraordinary to me. It's those new receptors we were talking about are not only regulating the body clock, but they're also plugged into those bits of the brain that increase levels of alertness. And so what you want to do is minimize light exposure half an hour before you go to bed. What do we do before we go to bed? Well, we stand in a brightly lit bathroom, looking into the mirror, masses of light, cleaning our teeth. So from now on, promise me, what you're going to do is clean your teeth in semi-darkness. It'll enhance your ability to go to sleep. Try not to nap during the day. Certainly don't nap for longer than 20 minutes. because what that will do is drive back that sleep pressure we were talking about, which means it'll be more difficult to get to sleep at night. Seek out natural light, particularly in the morning, because what this does is adjust the body clock to a slightly earlier time. Morning light is when you really need to seek out light. And then, of course, avoid caffeinated drinks much after lunchtime, because that will interfere, as we talked about earlier, with that substance adenosine, which is building up the sleep pressure. Shift work represents yet another problem. You can be on the night shift for 20 years, but the body clock doesn't adapt to the demands of sleeping, of being awake at night. So anybody like to suggest why? Why don't we adapt to the night shift? I mean, if you fly from London to New York, the body clock adapts. So why don't shift workers adapt? Any thoughts? Would you like to shout out a question? I shall ask Jonathan if nobody puts their hand up. Natural light, exactly. So Jonathan, you're off the hook. So essentially what's happening is the night shift worker is working under fairly dim light in the workplace and then is experiencing bright natural light on the journey home or during the day. And of course, the clock will defer to the brighter light image of the brighter light world as being daytime and keep the body clock locked onto the external world. And so what we see in night shift workers is that when they're trying to function, their body clock is in the resting state. So you can't possibly expect a peak performance from these individuals. But it's even worse because, of course, when they're trying to sleep during the day, here, even though the sleep pressure has built up and they need to sleep, the body clock is saying, hang on, no, this is daytime. You need to be awake. And so the quality of sleep that many night shift workers get is usually very poor indeed. Okay, this is a wonderful clip I found, which is this was an article that was put up on a notice board for night shift workers, and so it said tips for married shift workers, and somebody had written underneath, "Don't work shift work." Okay, so why? What are the consequences of all of this? So let's now consider the impact of sleep and circadian disruption, which we've called SCARD. It sounds a bit like a Bond villain. So let's look at SCARD on our ability to perform. So studies from the United States have shown that in the USA, 100,000 car crashes every year are related to sleepiness. This is driving, of course, when the body clock is saying you should be asleep, and also accumulated fatigue. In 1989, of course, we had the Exxon Valdez, an accident that caused incredible ecological damage, and the accident investigators determined that fatigue was a factor in the crash. Everybody still thinks that the captain was drunk. Actually, he was drunk, and he was asleep in his cabin, and the people driving this boat were deeply fatigued and tired, and were being told, don't move the ship, don't take it into the reef, and they just couldn't process the information, and this thing hit the reef. This disastrous air crash in 2010, the Air India Express, accident investigators said that the pilot could be heard snoring in the cockpit voice recorder shortly before the disastrous landing, which killed so many. And of course, the worst nuclear disaster ever, 1986, the International Nuclear Safety Group determined that human error related to sleep deprivation was a factor in the accident. What was going on is that they were doing some sort of experiments, and essentially they were sleep deprived and disinhibited, and essentially weren't really fully aware of what they were doing, and so that led to this appalling nuclear incident. The problem is that the tired brain, when it's forced out of bed by the alarm clock, then resorts to stimulants. Caffeine. We've already talked about caffeine blocking those adenosine receptors and keeping you awake. The really naughty, tired brain will also use nicotine, and that's very effective at allowing you to stay on task. The problem is that the waking day is increasingly fueled in the developed societies by these stimulants. And these stimulants, of course, last in the body for some time. Caffeine can last, it has a half-life of 5 to 9 hours. So a drink or a double espresso after lunch will certainly delay the time that you will go to bed at night. And if the day is fueled by these stimulants, you get to the night time and think, well, hang on, how am I going to get to sleep? Why don't I pop a few sleeping tablets or, in fact, resort to a substantial glass of whiskey? Now, they will indeed sedate you, but sedation by these substances is not the same as biological sleep. And so many of the benefits that you get for sleep that we were talking about earlier, enhanced ability to lay down memory, one's creativity, will be affected by these sorts of sedatives. And then, of course, to counteract the sedatives, you need more stimulants after the alarm clock has driven you out of bed. And this stimulant-sedative feedback loop has become the existence for so many millions of people, tens of millions of people all over the world. And a little factoid is that after oil, coffee beans are now the highest traded commodity on the open market, showing how, as a society, we've been utterly dependent and addicted to caffeine. There's a very important relationship between sleep, the guts, and appetite. So, for example, the hormone ghrelin, when released and hits the brain, will promote carbohydrate consumption and will predispose you to weight gain. Whereas leptin tends to decrease appetite and so predispose you to weight loss. And so you have this sort of seesaw of weight gain and weight loss, depending upon availability of calories and all the rest of it. However, what sleep loss does, sleep and circadian rhythm disruption, is hugely swing this seesaw to the ghrelin side and the predisposition of weight gain. Ev Van Kouter did some beautiful studies in the University of Chicago. She took healthy young males in their 20s, one group allowed to sleep 4 hours a night, the other group 10 hours a night should they want it. By the end of 7 days, their ghrelin levels were up by 28%. Their carbohydrate consumption had gone up by 35 to 40%, and their ability to clear glucose from the circulation was bordering on the diabetic. So even 7 days can have a big influence on that seesaw of the ghrelin-leptin appetite axis. No great surprise that obesity is much higher in night shift workers compared to day shift workers. There's also a very interesting relationship between the stress axis and sleep-wake. Of course, acute stress is critically important. Cortisol, the stress hormone, the fight and flight response, incredibly important. But with prolonged sleep deprivation, sleep and circadian rhythm disruption, as you have in many teenagers and many night shift workers, then drives this axis way outside of its normal physiological levels. So you find glucose, when you're stressed, is thrown into the circulation and insulin is decreased. So essentially, this predisposes you to glucose intolerant. Diabetes 2 is much higher in shift workers compared to day shift workers, and in tired and sleep-deprived people in general. Gastric acid secretion is up after sustained levels of stress, and of course, predisposes you to stomach ulcers. And ulcers are a much higher, taxi drivers, classic example of stomach ulcers, night shift taxi drivers. The one thing that we know about sustained levels of glucocorticoids, such as cortisol, is that they will suppress the immune system. And this suppression of immunity of the immune system has been linked to higher rates of infection and cancer in tired and sleep-deprived individuals. Blood pressure is elevated, cardiovascular disease is higher in stressed individuals with SCARD, and of course, under long sleep and circadian rhythm disruption and sustained levels of cortisol, you even have memory impairment. So the impact of SCARD, cognition and memory are affected. You're much more prone to substance abuse. You have metabolic abnormalities like diabetes 2. There's cardiovascular dysfunction, and there's suppressed immunity. Okay, let's now in the last few minutes try and pull some of these things together with a discussion of mental illness. We've known about sleep and circadian rhythm disruption in mental illness for a very long time. This is Emil Kraepelin, the father of psychiatry, who in the 1880s was talking about abnormal sleep in conditions that we would call schizophrenia today. And from diary data, here's Vincent van Gogh, I often think that the night is more alive and more richly coloured than the day. I put my heart and my soul into my work and have lost my mind in the process. So we've known for a very long time of the association between sleep disruption and mental illness. However, if we jump forward 100 years from the 19th century to the 1970s with the introduction of the antipsychotics, individuals or practitioners then said, well, schizophrenia and the bad sleep you get in schizophrenia is clearly due to the antipsychotics that they're taking, ignoring the fact for the previous 100 years there had been abnormal sleep without any use of antipsychotics. If the antipsychotics weren't blamed, then abnormal sleep was attributed to the basis of lack of work. So, and this is a quote from somebody who will remain anonymous, which is, my patients can't hold down a job, so no wonder they get up late, miss my clinic, and don't have friends. In fact, it was that statement in a lift in a hospital in London that got me into this whole area in the first place, because that just didn't make any sense at all. So where we were was that, yes, there is an association between psychiatric illness and sleep disruption, and it's probably due to, well, the social isolation or the medication. Felt deeply uncomfortable with that sort of statement. So we then, with my colleague Katharina Wulff, looked at schizophrenia and sleep disruption in some considerable detail. Here we have one of those patterns. We're measuring the rest activity. Here's midnight. And we've got 2 days plotted, so you can see the pattern. This is basically the nighttime, and this is the daytime when these individuals were active. And we see a beautiful pattern of rest and activity. And this is an unemployed individual, and who are statistically not different from employed individuals. In schizophrenia, it's absolutely shocking. And these next series of slides show different individuals. So we're seeing huge amounts of activity after 11 o'clock at night. These individuals are drifting, no ability to lock onto the light-dark cycle. Huge amounts of activity, then it breaks down completely, and it gets worse and worse and worse. This individual has no 24-hour patterns whatsoever. These are the worst sorts of rest activity profiles I've ever seen in, let alone a mutant mouse, but in humans as well. This individual we've followed for several years now, 4 years, and you see that she's very late, she's going to bed late, but she's also breaking away. She can barely lock on. She's normally sighted. There's nothing wrong with her eyes. She spent the whole of this year simply getting up later and later and later. It's absolutely smashed. These are appalling patterns. And we've now shown that these patterns are independent of the medication that the people are on, or indeed the fact that they don't have the social constraints of a job. We've also, as with others, of course, have highlighted, of course, the poor health that you find in mental illness, impaired cognition and memory, increased substance abuse, metabolic abnormalities, cardiovascular disease, reduced immunity. Where have we seen this before? Yep, the impact of sleep and circadian rhythm disruption. So one has to ask, to what extent in mental illness is the impaired cognition, the substance abuse, the metabolic abnormalities, and all of these others the product of sleep disruption or the primary mental illness? And yet one more connection, it's neuroscience. Ta-da! We talked about this earlier, and I asked you to remember this image. This is the generation of normal sleep, all of these various component parts, all these neurotransmitters. So what this tells us, of course, is that if you have a defect in a neurotransmitter system that predisposes you to mental illness, whether it be in dopamine or serotonin or acetylcholine, whatever, you're almost certainly going to have an impact upon sleep. And so what this all collectively led to was a different way of thinking about sleep disruption in mental illness, in that psychiatric illness and sleep disruption share common neural overlapping pathways. Of course, the sleep disruption will make the psychiatric illness probably worse, and the psychiatric illness will probably exacerbate the sleep disruption. So what's the evidence for that? It's still early days, but I just want to give you a few bullet points. So what about evidence for common neural mechanisms between psychiatric illness and sleep disruption? What we found is that genes that have been linked to mental illness are also having a big impact upon the ability to generate normal sleep-wake rhythms. So, for example, we've taken a gene that's been linked to schizophrenia in humans, mutated that gene in a mouse, and those mice have got smashed sleep-wake cycles, just like the patients. It's now becoming clear that genes that also drive normal clock and circadian rhythms also predispose individuals to mental illness. So there's a genuine mechanistic overlap between the two systems. What about sleep disruption and psychiatric illness? Well, and also within the context of common overlapping mechanisms. Remember what they were saying was that the psychiatric illness was simply giving rise to the bad sleep. So what we've been studying, this is Guy Goodwin in psychiatry, let's look at kids who are at high risk of developing bipolar disorder. And what's extraordinary, there's no clinical diagnosis of bipolar, but they're already showing really quite marked abnormalities in their sleep-wake profile. So again, more support for the idea of common overlapping neural mechanisms. And then the final thing about sleep disruption affecting psychiatric illness, Dan Freeman in Oxford has taken individuals with schizophrenia, partially consolidated their sleep-wake. And what he's shown is that he can reduce levels of paranoia in those individuals by 50%. Again, showing an absolute link between sleep disruption and psychiatric illness. Okay, why do I think this is all quite interesting? My final slide is summarizing it here. Why I think we have an area of really interesting investigation is what this is all doing is providing a better understanding of the brain and genetic mechanisms that give rise to, that are common to sleep-wake timing and neuropsychiatric illness. We're actually understanding the fundamental neuroscience of these systems by looking at them using a range of animal and human models. More importantly, perhaps, is we can use sleep and circadian disruption as a biomarker in the early diagnosis of neuropsychiatric conditions. If we can identify those individuals who are at high risk, then we have the chance of going in and maybe consolidating, or at least give the option of clinical intervention early on. And then finally, we can use agents and protocols that regulate sleep and sleep timing for the reduction of symptoms and hopefully for the improved quality of life in neuropsychiatric conditions. Okay, we finished. Let me just finish with a few more points. We discussed this world of 24-hour rhythms that, in a sense, 24-hour rhythms in our physiology and behavior dominate much of what we are and are driven by the brain and internal clock systems. And sleep is a complicated amalgam of multiple brain structures and neurotransmitter systems. We talked about the importance of the eye regulating the body clock and aligning internal time to the external world. And in the process of trying to find out these mechanisms, identifying another light sensor within the eye. And this is now having a big impact upon clinical ophthalmology. We talked about working against internal time with several examples, mostly within the context of teenagers, but we also touched upon shift workers. And essentially, working out of sync with internal time has real health consequences, which we are, as a society, largely ignoring. And then finally, this sort of new area where an understanding of the neuroscience of body clocks and sleep is allowing us to think about new ways of dealing with mental illness. Not only mechanistically, but also we can think perhaps of sleep as a new therapeutic target, consolidate sleep in these individuals, and hopefully you'll be able to improve the quality of life. But also maybe if you can go in early on, you can consolidate sleep. Maybe you'll nudge the developmental trajectory of these individuals out of a neuropsychiatric illness and perhaps delay onset or maybe avoid onset completely. And all of these sorts of topics that I've been talking about are now being studied within our new institute in Oxford, the Sleep and Circadian Neuroscience Institute. And with that, I'll finish and take questions. Thanks very much for your attention.
Okay, so if anyone does have any questions, there are 4 microphones positioned around the auditorium. So if you could come forward to the microphone if you have a question.
Thanks. First question. Do we know whether we have a new prince or princess?
No.
It's a boy. Ah, well, you heard it first here. Fantastic. Okay. Any questions?
Yeah. Over mic 3, over here.
Okay.
Keith.
Hi, Keith.
I'm just curious about Scandinavian countries, or ones that are very far north, where over the 6-month cycle you get almost total sunlight, or then alternatively in winter total darkness.
Yeah.
How have those people adapted to it, and the impacts on health for them?
It's a really interesting question. What the people of Tromsø do is they use light boxes. Huge numbers actually get a light box, and they will try and actively consolidate their sleep-wake cycle by exposing themselves particularly to morning light. And so they're aware of it, and they take countermeasures. What's really interesting, and of course what they're also trying to push back is a condition like seasonal affective disorder. Seasonal affective disorder has been shown to be enormously improved by the light box use. And it's been reported that as many as 30%, I think, of the individuals in Tromsø normally have severe, sad seasonal affective disorder, which is to some extent alleviated by these light boxes. But those are Europeans that have been there for relatively sort of few thousand years, maybe. The Inuit have been, of course, in these northern latitudes for 20, perhaps as much as 30,000 years. And I am told that they don't show any symptoms of seasonal affective disorder or are affected by this absence of a time cue for big chunks of the year. And I think that's really interesting. But to my knowledge, nobody has actually studied the Inuit in any sensible way. I was talking to a colleague in Toronto fairly recently, and they're very keen to try and work with these individuals. But I don't think there's any good data out there sort of that will help us explain why they don't have this circadian rhythm disruption.
Could they look at other animals then that live in the same area?
So what about the Arctic reindeer, for example? Karl-Arne Stokken, in Tromsø, has looked at this, and he's shown that the Arctic reindeer, during the constant light and the constant dark of summer and winter, turn their clocks off. They show no 24-hour pattern whatsoever. And the argument in the winter is they'll forage whenever the weather conditions will permit. And so there's no point in actually having a clock. And there's no point unless you can align the internal world with the external world. So 24-hour rhythms are actually disrupted in those Arctic species. Whether it's true for all Arctic species, don't know. But he's got some lovely work on that.
Thanks very much.
Thank you. That's it? Oh, there's somebody over here. Number 4.
Hello, thanks very much for a really interesting talk. It was really informative. Had a quick question. Do you believe that the larks and the owls are inborn and you have a genetic predisposition to prefer either the morning or the nighttime?
There's certainly a genetic predisposition to morningness and eveningness, and some lovely work has come out of the University of Surrey in that regard. But there's this genetic predisposition, but that will also be coupled with developmental changes, as we were discussing with the teenagers. Those events during puberty will tend to make you go to bed later and get up later, even whether you're a lark or an owl. So that will be imposed upon that. Those hormonal changes will be imposed upon sort of a genetic basis for morningness and eveningness. And as I say, we've got some very good examples. That familial advanced sleep phase syndrome, absolutely classic. A tiny change in one of those clock proteins has been linked to this real change in the morning-evening disposition of those individuals. So yes, there is a genetic basis for it.
Okay, thank you.
Thank you. Oh, number 3.
So what about aging? So there's huge amounts of disruption of regular sleep patterns, and nobody's really reported massive psychiatric ailments across that population.
Yeah, I wouldn't want to give the impression that disrupted sleep causes psychiatric illness. I'm just saying that if you have a defect in a neurotransmitter, let's say that's common to both, then no great surprise that in psychiatric illness we'll see disrupted sleep, and then the disrupted sleep will exacerbate the psychiatric illness. But going back to the aging, which I think is really interesting, because we've got relatively little idea about what's going on. I mean, stuff that we've done in animal models has shown that the strength of the light signal getting to the clock isn't as pronounced in aged mice compared to young mice. So maybe part of the problem is that the clock is less sensitive to light, and therefore it's more inclined to drift. And some lovely work from from Aus van Summeren has gone into the nursing home and increased the amount of light during the daytime and darkness at night. And he's been able to stabilize to some extent the sleep-wake pattern of these individuals. And in a population of mildly demented aged individuals, cognition went up by 10%. So providing a much more robust light-dark cycle in the aged may be actually of huge benefit.
On that,
Yes.
are the photosensitive RGCs more subject to loss over a normal lifetime?
That's really interesting. There are arguments both ways. There's a bunch of studies from animal models suggesting that they actually may be more resistant to retinal damage than the other ganglion cells. Other studies suggest that that may not be the case, but it's a really very interesting issue. What we have shown is that in aged mice, the projection from those ganglion cells to the brain is substantially reduced. Whether the cells are going or whether the projection from those cells to the brain is going, don't know at this stage. But you know, it's really interesting. I mean, when you think about the importance of sleep-wake in the elderly population, we have very little idea of the neuroscience behind disrupted sleep. There's another question, number 4.
Yeah, I was just wondering, you were saying that in the absence of any normal light input, a healthy person will default to a longer than 24-hour
Yes.
cycle. I think I've heard figures of 26. I don't know if that's true or not. But,
It depends again on who you talk to. I mean, the early
Okay.
data from Germany suggested about 25 hours. I think the consensus these days is between 24 and a half, is about 24 and a half, perhaps 24 hours, 10 minutes, 24 hours, 30 minutes. It's in that window there. Yeah.
So my question then is, given millions of years of evolution, what's the teleological advantage to a 25 as opposed to a 21-hour cycle? Is there a good reason for it? Or?
Yeah, and actually, within any population, you get quite a broad distribution of sleep-wake, as we've discussed on the basis of the genetics and hormonal. And it raises why the clock isn't 24 hours in any species. And what I'm told is, and I can't pretend to understand the models, is that it's much easier to lock a 24-hour rhythm, a rhythm that is not exactly 24 hours, to a 24-hour rhythm. The modeling suggests that if you have a biological 24-hour clock and an external 24-hour oscillation, it's much more difficult to align them. So with a clock that's slightly not 24 hours, it's easier to sort of shift it to the 24-hour light-dark cycle. Because, of course, the 24-hour light-dark cycle only at the equator is going to be the same. Dawn and dusk is moving backwards and forwards, and of course the clock needs to be nudged backwards and forwards throughout that cycle. And as I say, it seems that a slightly non-24-hour oscillator is easier to lock onto a 24-hour oscillation under those circumstances.
Thank you.
Thank you.